Okay, good afternoon, students. In previous class, under the chapter Biotechnology, Principles and Processes, in this particular chapter, we have discussed regarding the features that are required to facilitate cloning into the vectors. What are those features that are required to facilitate cloning into the vector? And still for so far, we have studied three features. First one is origin of replication, that is ORI. Second one is selectable markers. Third, we have discussed the cloning sites. Okay, and we have discussed the cloning vector also, PBR 322, the structure, the diagram, and its explanation about the structure of PBR 332. Okay, in today's class, we are going to discuss the next point regarding the features that are required to facilitate cloning into the vector. The fourth and last feature, that is vectors for cloning genes into the plants and animals. Okay, you all know about some characteristics of what we are going to call that as a genes, which are what means there are some pathogens. Pathogens are nothing but what? The disease causing organisms. Okay, disease causing organisms, and what they are in those pathogens, the particular pathogens are there, they are going to convert the normal cell into the cancerous cells or a tumor cells. Cancerous cells or a tumor cells. Okay, we are going to discuss such two pathogens in today's class. That is helpful for the means we are going to study those under the vectors for cloning genes in a plants and animals. Right? The first example, still possible, you all are studied regarding how we are going to transforming the genes, okay, into the water, E. coli bacteria. Then you are going to transfer that into the particular host cell, plant or a animals, with the help of what we have studied regarding the plasmid. Plasmid, it is act as a vector. Okay. Now here some vectors, vector for a cloning genes. We are going to study vectors for a cloning genes in a plants and animals. The example one that is Agrobacterium tumefaciens. Agrobacterium tumefaciens, it is a pathogen. It is a pathogen for a several dicot plants. It is a pathogen for a several dicot plant. And here a piece of eDNA is going to transfer. A piece of eDNA is to transfer. I written here, the piece of eDNA is transferred the normal cell into the tumorous cells. Whatever the Agrobacterium tumefaciens is there, this is the pathogen for the several dicotyledon plants. In that plant, what it is going to do? It is going to transfer a piece of eDNA, and that eDNA is going to convert normal cells into the tumor cells. In the sense, it is going to cause the cancer, like cancer only. Tumor means the tumors are going to grow. Understood? And it is in the case of what? In a plant. Another example is there. We are going to study that retroviruses in animals. These retroviruses also have ability to transfer the normal cell into the cancerous cells or a tumor cells. In the sense, tumor cells, and they cause the cancer in animal. And that is what we are going to call as a cancerous cells. What it is? This is their normal, what we are going to call that as a nature. And you all know about the plasmid. We used to modify such genes, and we are going to suppress which gene is we are not interested, and we are going to promote the growth of what we are going to call that as the gene which we are interested. By doing that, what? Cloning site. We are going to choose the cloning site. Then we are going to add the alien piece of DNA, and due to the insertion, what we are going to call that as a insertion, inactivation, and alternative formation of what we are going to call that as a suitable markers. Okay, these all things we will study. So these two things, in the sense bacterium tumefaciens, that is a pathogen which is going to cause the tumor cells. It is going to convert the normal cell into tumor cells in a plant. And retroviruses are they are also having the ability to transfer the normal cells into the cancerous cells. Now, by better understanding of the art of delivering the gene by pathogen into the eukaryotic cell, knowing the art of delivering the genes by the pathogen into the eukaryotes, what we are going to do, we are going to modify TI in the sense Agrobacterium tuberous, Agrobacterium tuberculous, Agrobacterium tuberculous pathogen, which are going to cause the convert the normal cells into the cancerous cells or tumor cells. That is TI plasmid. TI plasmid of Agrobacterium tuberculous. It is modified into the cloning vector. We are going to modify that into the cloning vector, in which is no more pathogenic to the plant. The normal one is pathogenic to the plant. We are going to modify now this TI plasmid. This is the TI plasmid of Agrobacterium tuberculous. We are going to modify that by using the biotechnology, and that time, that is no more pathogenic to the plant, but still used to mechanism to deliver the interstate gene into the vector of variety of plants. But still we are going to use that plasmid. The plasmid which is having the what we are going to call it as a eDNA or TI plasmid. Normally it is what we are going to call that as a having the ability to transfer the normal cells into the tumorous cells. But now what we are doing, by using the biotechnology, we are going to modify this TI plasmid of Agrobacterium tuberculous in such a way that this TI plasmid is no more harm to the plants, but still it can use to transfer the interstate gene into the variety of plant, that is the vector for a cloning gene in a plant. Which one? That is TI plasmid. I hope it is understood. Now next one is similarly the retrovirus have been modified and used for delivering the interstate gene into the variety of animals. So this is about we are going to write regarding the vectors for cloning genes in a plant and animal. Whatever I written still possible up to here. Agrobacterium tuberculous and retrovirus. Normally they are harmful for the plants and animals. They are going to cause or they are going to convert the normal cell into the tumorous cells. But when we are going to modify them, that plasmid, TI plasmid or retrovirus plasmid, and that time we are going to suppress the harmfulness to the particular plants or animals, but we are going to use that plasmid to transfer the interstate gene into plants and animals respectively. And I written the name of those are TI plasmid and retroviruses, modified retroviruses. Okay, this is about what vectors for cloning genes in a plants and animals, and this completes regarding features that are required to facilitate cloning into the vector. First one is origin of replication override, selectable markers. Third one is cloning site. Fourth one is vector for cloning gene in a plants and animals. Right? And you all know about the structure of PBR 322. Okay, and you used to remember that diagram. So many times we used to ask you draw a neat label diagram of plasmid PBR 322. Okay, right. This is about what the features that are required to facilitate cloning into the vector. Now we are going to move. You have modified the cloning vector, not at all. But how we are going to transfer that plasmid into the bacterium, E. coli bacterium? It is not an easy job. Okay. For that, what we used to do and what are the techniques are there to transforming this modified plasmid? The modified plasmid which have the interstate gene. Whatever we are having the interest means we have interested in gene. That gene we are going to clone into the vector. We are clone that gene into the vector. Now what we are going to do? We are going to transfer that vector into the bacterium, E. coli bacteria, or we are going to transfer that into the host cell. Okay, is the recombinant plasmid aitu. A recombinant plasmid na em madbeku host cell naga introduce madbeku. How we are going to introduce and what are the methods? For that we are going to study the next point. We are going to call that as a competent host. Competent host in the sense ಯಾವ್ದೊಂದು ಹೋಸ್ಟ್ ಇರ್ತೈತಿ, ಯಾವ್ದು ರಿಸೀವ್ ಮಾಡ್ಕೊಳ್ಳೋದ್ರೆ RDNA ಅದು ಕಾಂಪಿಟೆಂಟ್ ಇರಬೇಕು. ಕಾಂಪಿಟೆಂಟ್ ಇನ್ ದ ಸೆನ್ಸ್ ಇಟ್ ಈಸ್ ಏಬಲ್ ಟು ರಿಸೀವ್ ದಟ್. Whatever we are going to putting the RDNA into the host, it has able to receive that. So again another point is the competent host or for transformation with the recombinant DNA. For transformation with the recombinant DNA. ಈ ಕಾನ್ಸೆಪ್ಟ್ ಮೇಲೆ ಬಹಳ ಸಲ 3 ಮಾರ್ಕ್ಸ್ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದರು. What are the different types of methods which are used for the competent host in a recombinant DNA technology? For this, there are the several what we are going to call that as a techniques. What are those in the sense? You all know about the DNA is hydrophilic molecule. DNA is hydrophilic molecule in the sense it is water loving molecule. So that what is going to happen? It cannot pass through the cell membrane. Okay. What I told you, DNA is hydrophilic molecule. So it cannot pass through the cell membrane. So we have to make. That's what we have to make the bacterial cell competent to take up the DNA. ಹಾಗೆ ಮಾಡಬೇಕಾದ್ರೆ ನಾವು bacterial cell ನ ಒಂದು ಈ ರೀತಿ ಅದನ್ನ ನಾವು ತಯಾರು ಮಾಡಬೇಕಾಗ್ತದೆ. That is competent to receive the DNA. And now what are these techniques ಅಂತಂದಾಗ there are 3 to 4 techniques are there which are helpful to make bacterial cell competent to take up the DNA. Okay, I used to write here what are those. Okay, we are going to discussing now how we are going to introduce the rDNA into the host cell. And I told you the DNA is hydrophilic molecule, so it cannot pass through the cell membrane. So we have to make the bacterial cell competent to take up the DNA. How we are going to make? In that, first one is we are going to go for the what? For this, we are going to do the treatment. We is to treat the bacterial cell. Okay, right? Treating them. Whom we are going to treating? Bacterial cell. Bacterial cells are treating. Bacterial cells are treating with a specific concentration of divalent acid. This is very important. Divalent cation in the sense such as calcium. In the sense, you are going to treat the bacterial cell with a specific concentration of divalent cation such as the calcium. So that what is going to happen in the sense, this increase the efficacy efficiency. It increase the efficiency with DNA. Efficiency with DNA enters into the efficiency of DNA enters into bacterial cell. When we are going to treat the bacterial cell with a specific concentration of divalent cation such as a calcium, that time it increase the efficacy of DNA enter into the cell through the pores. Pores present on the cell wall of bacteria. Cell wall of bacteria or cell wall of bacterium. Understood? So this is one thing. Now, first what we are going to do, we are going to go for the bacterial cells are treating with the specific concentration of divalent cation such as calcium. Then, what is going to happen? That can increase the efficacy of DNA enter into the bacterial cell through the pores present on a cell wall of bacteria. And what is the procedure you have to follow for here? What you are going to do? You are going to do first rDNA and what we are going to call it as rDNA. rDNA is what is we are going to incubate rDNA, then forced into such cells by incubating these cells. In the sense, rDNA and bacterial cells are incubating. What I am telling you is here, first you did this. What? The bacterial cell wall is made in such a way that means you are going to treat that with the what? Calcium, that is what? Divalent cation. Then those bacterial cells and rDNA are incubating, okay, on ice. Incubating on ice. In the sense, what you are going to do? You are going to put together. What are those? The cells which are treated with the divalent cation and rDNA on a ice. That is what we are going to call it the process of incubation. Then you are going to go for giving 42 degrees centigrade, degree Celsius, 42 degree Celsius heat shock. Used to go give the 42 degrees Celsius heat shock, SHOCK. Then again we are going to put those both rDNA and what we are going to call it as a rDNA and B cells on a ice. You understood? First on ice we are going to incubate, then you are going to go for the 42 degrees centigrade heat shock, then again you are going to put on a ice. Again you are going to put on a ice, back to the ice. That is enable to bacteria to take up the, and this helps bacteria to take up our DNA. Okay, I hope it will be understood. What I told you, competent host hen module. What I told you, the DNA is hydrophilic molecule. It cannot pass through the cell membrane. We have to make the bacterial cell competent to take the DNA. Andre now bacterial cell hang madಬೇಕು ಅಂತಂದಾಗ competent to take the DNA. How we are going to make the bacterial cell competent to take the DNA? First method is used to treat the bacterial cell with a specific what concentration of a divalent cation such as calcium. Okay, if you are going to treat in such a way that time what is going to happen? The DNA is going to enter into the bacterial cell through its pore. Okay, now what is the procedure? After that, what treatment of divalent cation, the bacterial cell and recombinant DNA, whatever we have prepared with earlier techniques. What are those? Origin of replication, another selectable markers, then cloning site. ಇವೆಲ್ಲ ಮಾಡಿ ನಾವೇನೋ recombinant DNA ಮಾಡಿರ್ತೀವಲ್ಲ ಅದನ್ನ ಮತ್ತೆ ಈ ಒಂದು ಟ್ರೀಟ್ಮೆಂಟ್ ಮಾಡಿದ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಎರಡು ಒಂದೇ ಕಡೆ ಕೂಡಿಸಿಡು. That is what we are going to call as a rDNA and bacterial cells are incubating. Where we are going to incubate? On a ice. We are going to incubate on a ice. Again ಆಗ್ತಿದೆ ಅಂಟಿ rDNA ಆ ಒಂದು ಏರ್ಪಾದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹೋಗು ಟ್ರೈ ಮಾಡ್ತೀವಿ. Then afterwards we are going to give 42 degrees centigrade heat shock, and again we are going to put these rDNA and bacterial cells on a ice, and after doing this process, that time what is going to happen? The bacterial cell is competent ಅಂತಂದಾಗ ಏನು ನೀವು ಕನ್ನಡ ಏನಂತೀರಿ? It is made itself to take up the DNA. It is going to made itself to take up the DNA. This is the first what we used to call that one as a bacterial cell. We used to make it competent to take the rDNA. Understood? This is the first one. Now second one we are going to discuss, that is, okay, second one we used to discuss, that is, this is the first one. Second one is micro injections. Micro injection in the sense we used to inject rDNA directly into the nucleus of animal cells. rDNA into the nucleus of animal cells. Okay, what I told you, rDNA is directly injected into the nucleus of an animal cell, of an animal cell. This is the second what the method is to make the bacterial cell competent to take the DNA. Understood? Okay. Third method is we are going to call it as a biolistics or gene gun. Biolistics or gene gun. What are these biolistics or a gene guns? Biolistics or a gene gun are what we are going to call that one as a these biolistics or a gene guns are suitable for transforming rDNA into the plant cells. rDNA transfer into the plant cells. Here, what is going to happen? These biolistics or the gene guns bombarded. These guns are going to bombarded BAM, BAR, DED, bombarded with high velocity. Bombarded with high velocity microparticles of a gold or tungsten, gold or a tungsten, gold or a tungsten coated, gold or a tungsten coated with rDNA. Coated with rDNA are called as a biolistics or a gene guns. Biolistics or gene guns are nothing but there are some what microparticles of a gold or a tungsten coated with the rDNA. We are with high velocity. We are going to bombarding on a plants. Bombarding in the sense what? Like this. Okay. So that they are going to insert into the plant cells. Insert into the plant cells. The plant injection direct injection so that we are going to use the bombarding technique. In the sense, biolistics or a gene guns, these are microparticles of a gold or a tungsten coated with the rDNA, and we are going to introduce them into the plant cell with a high velocity, bombarding with the high velocity. That is what biologistic to the gene gun. In such a way that we are going to introduce the rDNA into the plants. And last one is disarmed pathogen vector. Disarmed pathogen vector. DISRMED. Disarmed pathogen vector. In disarmed pathogen vector, okay, the last method is disarmed pathogens. Last method is disarmed pathogen vector. Disarmed pathogen vector in which which allow to infect the cell and then transform the rDNA, and it helps to transform the transfer or what we are going to call it as a, which is helps to transfer the rDNA. Okay. So this is about what the competent host for a or competent host in bracket what I written for a transformation with recombinant DNA. Transformation with recombinant DNA. I told you DNA is a hydrophilic molecule. It cannot pass through the cell membrane, so we have to make bacterial cell competent to take up the DNA. The methods: first we are going to treat the bacterial cell with a specific concentration of divalent cation such as calcium. This increase the efficacy of DNA enter into the bacterial cell through their what we are going to cell through their pores which are present on a cell wall of a bacterium. Afterwards, such splitting bacteria and rDNA are incubated on a ice and it is heat shocked by the 42 degrees centigrade. Then you are going to put again back into the ice, and this helps to bacteria take up the rDNA. That is the first method. Second one is microinjection. Here inject the rDNA directly into the nucleus of an animal cell. That is simple. Third one is biologic or a gene guns. Gene guns or a biologics are nothing but the microparticles of a gold or a tungsten coated with the rDNA. They are bombarded on a plant cells or bombarded on a plant with a high velocity. That is nothing but biologics or the gene guns. And last one is disarmed pathogen vector. Is nothing but the disarmed pathogen vector is allowed to infect the cell, and through the infection of cell, it allows to transfer the rDNA. So this is very important, and this is going to ask you for a 3 marks question. Write a note on competent host of bacterium to take up the rDNA, or write a note on competent host of a bacterium in a recombinant DNA technology. That time you are going to write these things. Okay, I'll stop here with this. In today's class, we will discuss regarding the vectors for the cloning genes in a plants and animals, that is TI, plasmid and retroviruses in animals. Then we will discuss we are discussing here regarding the competent host for transformation with the recombinant DNA. Okay, I am going to continue with in the next class with processes of recombinant DNA technology. Process of recombinant DNA technology. I am going to stop here. Thank you. I will continue next.